Hi, Science 10. In this video, we're going to talk about the scientific method when, well, yeah, the development of cell theory, some of the background, some of the key scientists, and yeah, how the scientific method resulted in us developing and understanding the cell theory. Ultimately, science is, well, it's about this. Take a look at these two photos. Can you spot the difference? Take a look. What you might have noticed is the one the lawnmower's plugged in, and it'll therefore work based on observation, right? You know that from observations. Science is ultimately about observations that can be made over and over by not just you, but by others. For example, maybe behind this forest, people think that there's Sasquatches, giant, hairy, like human creatures. People may have seen something like that, but if we can't consistently go out in the forest and find them, other people, then it's probably just bears. Bears standing up on their hind legs. They're big and hairy, and they can actually walk around like us sometimes. So it's probably that. That's the most logical explanation. But again, we need evidence. You need observable evidence and controlled experiments. But yeah, let's look at that today in this video.
An intro there. The video is, of course, about how cell theory developed, ultimately through keen observations and observations that could be repeated over and over, and controlled experiments. We see a couple of those examples right here. That actually wouldn't be the first microscope, but that's a compound microscope we're all familiar with. But yeah, let me share my screen. Let's get underway with the notes. Again, make sure you've got your notebook and be pausing the videos and taking down the information, the key information, because what we show you is what you need to know. I'm just trying to get my screen to share properly, so I'm sure it will in a moment. There we go. All right. So quickly, these are all the videos you're going to need to do all of these assignments, parts of a cell assignment, cell organelles assignment, microscope assignment, and the Chapter 7 assignment. So once you finish this video, make sure you're finishing all of those. There's your readings in our notes. This is where we are in the course text. Again, it's optional, but we've got a copy of it in the Moodle course. You're definitely going to be wanting to read the note package that we have in the Moodle course. But yeah, let's begin. So the development of cell theory. Ultimately, we all know that this mold right here, it comes from tiny mold spores. They are invisible, of course. These mold spores, well, they're cells, and life comes from life. Mold comes from mold. We know that. In the past, folks didn't. And ultimately, this video is about the key scientists. So Pasteur, Francisco Redi, Robert Hooke, and there's more than just them, but these are the three we're going to look at. Some of the key scientists, what they did, the scientific method. So basically, evidence and controlled experiments. And evidence doesn't just mean one person sees the information. It means many others and over, and we can see it and review it. And of course, controlled experiments, and we'll look at that. And it led to the development of cell theory, which you need to know the 3 points of cell theory. But yeah, let's start with, life does come from life. This picture here, we see a bunch of zebras. Now, of course, this isn't anywhere near where we're at. It's the Serengeti Valley in Africa, but a fantastic place full of lots of life forms. They all come from other life forms. Zebras come from zebras, elephants come from elephants, the trees came from other trees. That's something that we just take for granted that we know, but it was not fully understood even a couple hundred years ago. And of course, cells, microscopes weren't readily available, and people didn't know about microscopic things like cells and microorganisms. So there was a lot of interesting concepts. So what did people think in the past? Let's take a little bit of a look at that right now. Before we knew about cells, the thought was, it's called spontaneous generation. Life can arrive from an active principle. And of course, that sounds fancy, that word, but it literally had no meaning. So in the past, this is of course not what we think, and we in fact know this is incorrect, but the past, people felt that life comes from something called an active principle. Again, this word sounds neat, nice and neat, but it has no meaning. Spontaneous generation, that sounds like magic. So literally, sweaty underpants plus wheat husks, wait 21 days and you get mice. This is literally what was written down and what people thought. Well, Francisco Redi comes along and basically says, maybe let's change this idea. He used the scientific method to find out if there is some active principle in the air, or is there something else? So let's take a look at what Redi did. Flip my slide. All right, so can life arise just out of a pile of towels? Well, he conducted controlled experiments to prove if flies came from other flies. That was his hypothesis. I mean, until you do an experiment over and over, this is an idea. Flies come from other flies, or life comes from life. Not the air, not some active principle. You can notice right here how he created a control group, basically just open to the air as normal and just wait over time. And of course, you know, maggots are going to form and, oh, maybe it came from the air. Well, if it came from the air, then this is your the experiment that he designed. You know, a little filter here. Air can come in, but the flies can't. And then, of course, a fully sealed jar. The fully sealed jar, nothing happens. And the same with the jar that has open to the air, because flies come from flies. The little maggots don't just actively, spontaneously generate out of nothing. So this is an example of a controlled experiment. And ultimately, we are controlling and observing. And controlling and observing in a way that others can repeat what we've done. All right, so let's look at another controlled experiment by Louis Pasteur. You might be familiar with something called pasteurization, that basically, you know, our milk isn't spoiled if it's pasteurized. Well, let's see where that came from. So Louis Pasteur, again, it's the whole idea that you know, the broth, oh, my broth spoiled, you know, my soup spoiled, must be an active principle. Well, Pasteur figured maybe it's microorganisms. So this is basically, and you can see there's a few typos in here, but essentially let's just, number one, let's heat the broth, let's boil it. This is what we do when we pasteurize milk or pasteurize orange juice. We heat it, we boil it. That kills the microorganisms, because getting sick with deadly bacteria is really not a good idea. Let's try to spell the word microorganism correct. So, and yeah, write this down with me. I'm going to heat it. Secondly, and this is how it's totally controlled, we're going to place the broth that's boiled. So in other words, it's got no microorganisms, or at least we assume that they're dead, in an S-shaped flask. Take a look at that flask. It is open to the air, but look at that curve, especially down here. Microorganisms are going to get trapped in there. That's the key design. That's the key inspiration, and that's Pasteur right there. A drawing of him. I didn't make that drawing. S-shaped flask. His classic S-shaped flask. Lets in air. Again, there is no active principle, but oh, maybe there's some active principle in the air. No, there's not, because the air could easily get in there. So air can come in. Comes in, no problem. But microorganisms get trapped. Ta-da. Waited a while, the broth did not spoil. So, you know, basically third step, wait 24, 48 hours, etc. You know, meanwhile, he'd have some broth that isn't boiled, you know, sitting on the side open to the air. And yeah, it's the broth that isn't boiled is spoiled. Hey, that rhymes. But the one that he put in his S-shaped flask didn't spoil. Okay, remember he had a hypothesis. I think there's microorganisms. I think there's actually little tiny, living things that get in there, and they like to eat just like us. And they're totally reproducing, and yeah, they make a mess of the whole thing. Therefore, there's microorganisms. Now, we need a little bit more evidence than just this, but this certainly helps provide some of that proof that there are microorganisms. It's a pretty good controlled experiment. Can be repeated again by others over and over. That's what science is. Experiments that can be repeated over and over. They get the same results. They don't get different results. They get the same results over and over. But we need more evidence, more observable evidence. So that's where Robert Hooke comes in. So let's look at Robert Hooke next. Again, pause the video, take the notes, make sure you know about the two scientists we've talked about so far. So Robert Hooke, back in about 1600, so a while ago, 30-time magnification, so a simple microscope, right? A simple lens, basically. He's the first to see and document and name cells. So where did the name cells come from? Robert Hooke. Now, of course, his microscopes led to better and better microscopes that eventually allowed us to actually see microorganisms, see the quote-unquote the invisible. No, there's no active principle. There's things. They're living things. Cells come from cells. They like to eat, and they will make our oranges moldy, and they will spoil our broth. And yeah, so these three scientists come together, and ultimately, here's the scientific method for us that these three scientists show us. And it's not really number one, number two, both of these together. You got to have controlled experiments. Careful, detailed experiments that others can reproduce, combined with observable evidence, and not just, oh, I saw a hairy creature outside my yard. No, like it needs to be other people and can be observed over and over again. Doesn't mean that we go and we still can do discovery and try to find new things, but those are all hypotheses, and that's okay. And in fact, it's very important to have that in science. But the scientific method itself, if we're truly doing the scientific method, it can be repeated over and over, not just one person, many people, thousands of people doing these experiments over and over, making the same observations over and over, has led us to cell theory. So let's look at cell theory. And these are the three things you need to know about cell theory right here. First of all organisms are composed of one or more cells. So there's microorganisms or there's multicellular like you and I, and plants too. And of course, we know some details about those cells. Those cells, they've got organelles, and you need to know those as well. But ultimately, all life is made up of cells. The cell is the smallest functional unit of life. We know that inside of cells, there's organelles for sure. Right here, you can see the nucleus. The nucleus cannot function all on its own. It needs the other organelles. In fact, it works with the endoplasmic reticulum and the ribosomes to build proteins. Then the Golgi apparatus packages those proteins, and then through a process of exocytosis, the proteins are sent out from your cell to the rest of your body. Everybody works together on the cell team. If you break a cell apart, it's not going to work. The nucleus is not going to work on its own. The ribosomes aren't going to work on their own. So the cell is the smallest functional unit of life, and all cells come from other cells. There is no active principle. There is no spontaneous generation of life, and the only exception that we still don't fully know is when did life first start on Earth and how did that happen, but that's not covered in this course. You can certainly look that up. It's pretty interesting. There's some science behind it, but it's still not fully figured out. This is for us what we need to know. All organisms are composed of one or more cells. The cell is the smallest functional unit, and all cells come from other cells. So let's just take a quick summary for today's video. So we've got our observations combined with controlled experiments. Actually, these are some of the images from 1820s and 1830s, so there was clear evidence of cells, of microscopic life, and you know, that the air somehow doesn't have an active principle. There isn't something called spontaneous generation, that indeed cells come from cells, life comes from life, flies come from flies. Very quickly then, and a reminder where you're at. You're doing the readings that's in the book, that's in our Moodle course, right? Following those page numbers, completing these assignments. These are for marks, everything in the highlighted here on the right. You can do everything right up to this Unit 4 Quiz 1. Get them all done. Do the readings. There's also questions in the readings. The textbook is optional. We suggest you read it. We've got those textbook pages there, but definitely focus on our readings and, of course, completing the assignments. Okay, take care. Have a good day.